Awesome. So yeah, coming live, we've got Logan from Molecule, we've got Dr. Danny Barr from Tel Aviv, and today we're basically going on the second instance of our Researcher AMA series, where we're looking to kind of bring the community into the kind of diligence aspect of looking at the individual projects that are listed on Catalyst. Danny's project has just been listed on Catalyst. We can send through a link where you can go check it out and learn more. But today we kind of wanted to ask questions that get into maybe the things that you can't read from the materials that presented, kind of reading between the lines. And we have a bunch of questions that we would love to get answers to from Danny. So yeah, would like to welcome the both of them and give them a second to introduce themselves. So Logan, go for it.
Sure. Thanks so much, Benji. My name's Logan. I'm the Biotech Operations Manager here at Molecule. I have a background in biomedical sciences and was part of the team also supporting Danny and his project getting launched on Catalyst.
Awesome. Danny,
hey,
please.
my name is Daniel Barr. I'm a researcher at Tel Aviv University. I have a lab focusing on molecular biology of aging at the Faculty of Medicine. And I'll be happy to take any questions about the project, tell you about my journey in crypto, in science, in central science.
Let's start with the crypto part, just because I think it's quite rare that we find a researcher who has a MetaMask wallet. So why don't you tell us your crypto journey and kind of how that led you to DeSci?
Sure. So from my perspective, I found out about Bitcoin relatively late. It was like 2014-ish, early. And for me, it was late because I used to know about all the PGP wars and all of that with Zimmerman, and kind of lost track. So for me, that was relatively late, and I saw it. I was immediately hooked. Like, this is brilliant. What else is new in the field? Because by then it wasn't really that new anymore, and everything felt like pets.com to me. Except this one kid I saw on YouTube, he was talking about this Turing complete blockchain, and this was for me phenomenal. So I actually had the privilege of participating in the crowd sale of the Ethereum, and have tried to be a productive member of society since. Played with all the cool gadgets, participated in a few testnets for the merge, Sugi, and others. If you'll go to my ENS name, you'll see, you won't be able to see that Genesis address. You'll see Tornado Cash, pre-sanctions. Although I'm Israeli, I'm not subjected to US law. Still, I try to avoid touching that contract post-sanctions.
Makes sense. Yeah, you're definitely an Ethereum OG. Well, I guess what was your foray into the decentralized science space? How did you find out about that? And what were your first interactions there?
So decentralized science is kind of the name that's now very big in crypto, but it rides on open science, which has been a big push of the scientific community for a while. And there are many, many important projects, very successful, some even with the government helping, God forbid, outside. So now we have the ORCID, where it's kind of a repository that other bodies can interact with and generate claims about works of individuals. We have bioRxiv, which wasn't around a few years ago. Putting everything in open source, open code is becoming more and more of a standard. Sometimes for the big players, they let them get around that, which we shouldn't. And of course, once I saw VitaDAO trying to do their thing and support lifespan research, I wanted to see if I can help. I actually talked to them. It was probably a year or maybe even 2 years ago. And back then, universities are still universities. These things are hard and slow, and that didn't work out just on the technical side. But Molecule really brought the solution because the university doesn't know what a blockchain is. They have a blockchain center that took money from the worst crook they could. Look, literally, Tel Aviv University took money from the worst crook they could to start a blockchain center, and Once they could, they just closed it and erased the name. So it needs a company to sit between it and the blockchain and kind of do all of that. So in that sense, I think what Molecule did is brilliant. Yeah, and I'm always happy to try a new experiment.
So interesting, Dan. I feel like you're like full of kind of like secrets and stories here. We're gonna be uncovering some like Easter eggs along the way.
There is one on chain. Let's see if someone can find it. There's a nice Easter egg on chain.
Oh, okay. You heard it here first. But obviously, you know, yeah, you've been around in the space for a while. You were familiar with VitaDAO. You've come back, you know, to Molecule to launch your project, which is obviously what we're here to talk about today. So I'd like to spend a little bit of time asking you some more questions like about your project, and obviously trying to explain it as simply as possible for our audience today, which is always a challenge. But I'm curious kind of what started you down this line of research. So, you know, you're here with this project, AB4AD, focusing on Alzheimer's disease. You know, the basis of your project is linked to the pathogen hypothesis, which some people may or may not be familiar with. So I would love for you to touch on kind of like your journey into this research space and kind of why this project is so meaningful for you. And yeah, tell us any more exciting stories along the way.
Sure. So for me, this project is sort of a detour because you think of Alzheimer's as tightly linked to aging, but if you target Alzheimer's specifically, for people who come from classical study of aging, it's a detour because usually you say, well, I just address fundamental aging and all the disease will be delayed, including Alzheimer's. So that's completely true. But sitting at the Faculty of Medicine, and my lab is located in the Building of Dental Medicine, which is part of the faculty here. I read a paper, it came out fresh off the press, talking about this oral pathogen that is heavily studied in our building and its role in Alzheimer's. And they showed like really nice evidence. And if you haven't heard of the pathogen hypothesis before, don't feel bad. I haven't heard about it either before that. And I started looking into it, and there was really a lot of evidence for it. It was a surprisingly good amount of evidence, multiple studies, multiple groups across decades, across countries, across continents, really, really strong evidence to support it. And in that specific study, what I found interesting was that they saw the pathogen in brains of patients, but also in controls, which is what you see in other cases as well. You look for a difference. But there was one individual, a 102-year-old woman, the oldest in the study, and her brain was completely clean, like nothing there. It wasn't the lowest, it was 0. And I asked, well, 102 years is a long time. How did she get to 102 being exposed to the same pathogens as all of us? And that pathogen couldn't enter her brain. And that led me to kind of expand that and ask, well, okay, so we have these people and we all know them, and they get to really old age. And we can look at centenarians, supercentenarians, but also just regular people who get to the late 90s, and they're completely sharp. And they're with us, and they're focused, and their memory is fine. Why? They're exposed to the same pathogens, they live in similar environments. It's not some gradual process. It's Alzheimer's disease is close to a step function. It's not really, but it's close to it. You either have it or you don't. And then I thought, well, if it's pathogens, maybe they have a really good immune system that limits the ability of these pathogens either to get into the brain or to spread in the brain. And maybe I should spend a few minutes talking about the pathogen hypothesis here, right? Because the pathogen hypothesis of Alzheimer's isn't about this specific pathogen. It's something that we know for now over 40 years. And that is that if you look at the brains of Alzheimer patients, you find more than in brains of non-Alzheimer patients, specific pathogens. And if you go and you take a mouse and you inject it, not necessarily to the brain, you can inject, for example, that oral bacteria, you can inject it into the gums, it will infiltrate the brain and cause Alzheimer-like symptoms in that mouse. And the most convincing evidence for me is from a national-wide study in Taiwan. They have a very good database collecting information. It's a very organized Asian country, so if you don't want to be in the database, you don't have to. You just need to write a letter and send it. Out of the tens of millions of people on that island, about 1,000 people decided not to be in that database. So it's very, very comprehensive. So if at late stage in life, or after 50, I think, you get herpes, your chance of developing dementia in the next 15 years goes up 2.5-fold, which is huge. If the doctor prescribed you any over-the-counter medicine for that herpes, so ganciclovir or Zovirax or any of those, you go back to baseline. That, for me, was the strongest evidence this works in humans. Now, this also makes sense from a biological standpoint because more and more evidence shows that A-beta, those peptides involved in Alzheimer's disease, they are actually antimicrobial peptides. They help contain pathogens in the brain and prevent their spreading. But it also comes with elevated immunity. So your body tries to fight the pathogen. At the beginning, it's good. It localizes it to the brain. It prevents it from spreading. But it keeps fighting. And you have low-grade inflammation for many, many years. And that causes brain damage. And then your brain looks like a battlefield because your brain has been a field where your body fights that pathogen. And against herpes and against PG, and we don't know all of the pathogens. We have a few that we kind of like for this role. It looks like in many cases we don't win at the end. It's a prolonged battle that damages the brain significantly.
Thanks so much for the background on here, Danny. You said like so many interesting things that I would love to ask you about, but I would love to focus in a little bit more about something you said about, you know, getting herpes and getting exposed to the virus and how it significantly increases your chance of getting dementia years later. And if you get treated for it, you know, that risk drops. I think that's such a wild, you know, realization maybe for people to have. And I think, you know, we came across a statistic somewhere that was like 60% of people over the age of 65 have had some sort of gum disease. And gum disease is obviously linked to this pathogen hypothesis because, you know, if your barrier in your mouth is weakened, right? Like this is your first barrier, line of defense for the body, your physical barriers. If that's weakened, you know, the bacteria are able to enter the bloodstream, enter the brain. And this is kind of the link between it there. So in terms of like, you know, the impact that your research would have here for people, I mean, like we're talking large proportions of people. I think everybody also has had somebody in their lives affected by Alzheimer's disease. What are you hoping to achieve with this research that you're trying to do? And like, what kind of impact are you hoping it will have?
So what we're hoping to see here is that we are hoping to be able to identify that signature, that immune signature of those people that go to really old age with no Alzheimer's. And this is the best response, right? Your body reacts well and you're immune. Or you have a lower risk, at least, for developing that. And that signature will, of course, differ for every pathogen. On the other hand, there are people who have the pathogen. They already may have low-grade inflammation and they don't know it, or they're susceptible to it. Their immune response isn't optimal. It may be enough to kind of delay things, but it's not optimal, and they're at risk. So we want to identify those individuals, identify the pathogen, and say, well, actually, if you got infected with this pathogen, and maybe you don't know it, because many infections with herpes are non-symptomatic. You don't see any symptoms, but the pathogen is still there, and it's going to get to your brain. So those people may actually very much benefit from getting treated either with just simple over-the-counter, super safe antiviral medications. In cases of other pathogens, that might be a different type of treatment that's needed. Or as a much, much longer vision, can we look at the great response of the people that get to old age without dementia and mimic that and say, well, this is actually where the antibody needs to attack optimally to get the best response possible. And can we take that and give it to everyone, either by vaccine or by antibody treatment? So the easier part here is prevention, just to identify people at risk and say, you need to be treated for this to reduce your risk. And the harder long-term is a cure or vaccine.
So interesting. And, you know, I'm thinking here as well, like, you know, some people really may not even know that they have been exposed to the virus, right? Like they're non-symptomatic, they're going about their daily life, and yet they have been exposed to this like massive risk and increasing their risk profile. So obviously, I think here there's an opportunity for some awareness or like awareness campaign that can be done around it too, just so that people like have this on their radar. And, you know, when it comes to maybe people's awareness of the pathogen hypothesis or just, you know, good oral health in general, like, do you think it's really well understood by people, or do you think that there's a lot more work that needs to be done to get people kind of like up to speed caring about this specific area?
So let's break that in two. About the risk of exposure, don't worry, we're all exposed to it. Almost everyone is exposed to HSV, to EBV, to PG, they're ubiquitous. You go and you test the population and you get 98, 95, 97% people positive with a 5% error. So the question is, was that exposure something that, okay, occurred early in childhood and you reacted well, or is the virus laying dormant somewhere and the reaction is not optimal, and every time you're stressed, for example, for that, you get eruptions? Spreading a campaign that if you have any symptoms of Alzheimer's as an adult, you should treat it, not because of the source, but because of risk of dementia later on, sure, I would support that 100%. Oral health, any good physician will tell you first thing, not only because of Alzheimer's, the same pathogens, they're involved with cardiac risk. Just like you said, when you have gum disease, you increase the chance of bacteremia. You brush your teeth too hard, bacteria goes to the bloodstream. Some of these pathogens go to the aorta, increasing your risk of heart attack. Some of them can go and cause rheumatoid arthritis. Also, it's a complex immune thing. So we don't completely understand in Alzheimer's how long you need the exposure for the pathogen. It's possible that late in the disease, the pathogen is no longer important. It's kind of a self-fulfilling prophecy where you have damage causes inflammation, causes more damage, causes more inflammation. But early on, we know we can treat that. So yeah, oral health, any doctor will tell you, is super important, not only because of your mouth, but because of many, many, many other things.
Got it. Danny, as you were speaking, I think something just clicked for me, but I wanted to make sure that it was actually scientifically, factually correct. So one of the stats you brought up was that if you're infected with herpes later in life, then your risk of dementia goes up. But I think maybe the logical jump there is that when you're older, your immune system isn't as good at responding to pathogens and infections as when you're young. So if you're infected with herpes at a young age, you may develop the necessary immunity and the protection against not just the virus, but also dementia. But if you're infected later in life, you can't respond as well. Your immune system isn't as adaptable. And so it's less likely to protect you against dementia, per se. Is that kind of what you're getting at?
Well, so no, it is the virus. It's not just it's the immune system does it through the virus here, and we know that because the drugs to treat the virus don't restore your immune function. You're right, 50, your immune function isn't as good as it was in 18, but because we know that if you treat it with antivirals and antiherpetic medication, they're fairly specific to that family of viruses, so it's not killing all of the viruses in your body. So you see that. And also we have a lot of mouse studies. So, and even 3D models in human tissue where you can take and create mini brains really many, in petri dishes, and you inject the viruses, and then you see all of the hallmarks of Alzheimer's. You see the secretion of A beta, you see hyperphosphorylation of the tau. By the way, when tau is hyperphosphorylated, do you know the abbreviation for that in the scientific literature? Yes, you do?
It's like
No.
p-tau 205 or something?
Yeah, so neurofibrillary tangles, they're short for NFTs, but these are the NFTs you don't want to collect. You'll see it in the literature, that's a different. So yeah, I think here, people will always question causality. I think here the evidence for causality for these pathogens is very, very strong. So I can't give you numbers of how many cases are because of this pathogen and that pathogen and how many pathogens are involved in it totally. Maybe we'll be able to answer that. Study for multiple sclerosis, which is also, we've known for a while, it was initiated by some pathogen probably, and then it causes autoimmune system to run off. So there was a study with the same technology that we want to use that found it was EBV. It's like over 95% of cases of multiple sclerosis, it's EBV that starts, and poor immune response to that virus, because actually the body later to generate a lot more antibodies. They're just the wrong antibodies. And later these antibodies start attacking myelin, and you have an autoimmune response in the brain, which causes damage, which causes more autoimmune response. So here, I think we're very, very confident pathogens are involved in many of the cases. And I'm only talking about sporadic Alzheimer's. Familiar Alzheimer's, it's a different story, but the overwhelming majority of cases is sporadic.
Makes a lot of sense. So, Danny, one of the things that you mentioned, one of the terms, and I think a big part of this project is discovering signatures of these patients who have been infected, but show some degree of protection or immunity to developing dementia. What does this immune signature mean, and why is that important, and how do they differ between these two kind of clinical outcomes?
Sure. So the virus goes and starts infecting cells, and our cells have a defense mechanism. They display parts of the virus, the proteins of the virus to the immune system, and the immune system starts generating antibodies. Now, these antibodies, and this is the acquired, not the innate immune response, this basically the body throws the dice many, many times, generates random combinations of certain genes. And from that, it generates a huge repertoire of antibodies. Most of them are completely useless. They get filtered out. And by chance, you throw the dice enough times, you get a few that work really well for that pathogen. And then the body expands on those, goes in that direction, generates more variety from them. This is a random process. This is the reason why two people can go get infected with the same virus, their same age, their same conditions. One of them walks off perfectly healthy, the other dies. To the point where we used to think Ebola was always symptomatic, right? It's Ebola, you bleed from your eyes. And then they started looking at people who lived with people with Ebola in their houses and did not have any symptoms, and they found a large percent of them have anti-Ebola antibodies. They managed to get Ebola asymptomatically. So this is how good our immune system is. So that process is a random process. Everyone generates their own antibodies. And from my perspective, going and exploring the sequence of these antibodies is very, very difficult. It's possible today, but it's difficult. I rather look at their targets. What part of the protein is bound by each and every one of the antibodies? And we can do that. We have several techniques. The one that I want, hopefully, to get funded for is called VIRScan. We have another one that we do in-house with mass spectrometry, and another one with peptide microarrays. But basically, we take small chunks of each protein. We cut up the proteins into small chunks, and we see where does the antibody bind. And maybe your antibody binds here, and my antibody binds here. And from that, we get a signature. You have 25 antibodies that bind 25 different peptides, and I have 15 that bind 15, and some of them are better than others. Some of them can go and neutralize the virus. So if the antibody binds, that virus can no longer infect cells. So even if the body isn't so quick to remove it, well, it cannot do any damage in the meantime. And some are less good at their job. And maybe they market for the immune system, but they need the immune system to come and do something, and there's a lot of viruses, so it's a less effective process. So this is the signature. Exactly which peptides are recognized by the immune system.
Thanks a lot for explaining the signature. I feel like, as a non-scientist, maybe it's kind of abstract for people to wonder, like, okay, what does the signature mean? Like, what are you even looking at? But yeah, it's just the way, like, each cell will respond differently in its environment, just as, like, I would respond differently to maybe my colleague asking me a question versus like a family member asking me a question. I'm producing a different response. So a signature is also just the way of, the cell responding to its environment based on a certain stimulus. So obviously, for the research, really important for you to dig deeper and try to find these differences so that we can all get to 102 kind of dementia-free, like the woman at the beginning of your story. And just kind of thinking, long-term, obviously, you're very busy with your research. You're a passionate kind of like Alzheimer's researcher. You've got your pathogen hypothesis, and you're like, okay, I now have a platform. Like, I really want to scream from the rooftops and tell people, this one thing about, my research or research in general, or maybe life as a scientist, kind of like what is it that you want to leave people with here today?
I'm not really good at screaming.
Or what Easter egg are you going to leave people with later?
Oh, the Easter egg they can find on their own. It's not that. I think really we're at the incredible time where we have a chance to make a profound difference in human health and lifespan, and not to be too cynical and to look at all of the great advances that we have and that are coming. This project, other projects, there are many, many great projects. Most of them will fail. Some of them will do spectacularly well and help us all.
So good. And what would you say to people who are maybe thinking about becoming young neuroscientists? What are some exciting topics in the field today that you think people should really be focusing on, and kind of like, how do you inspire the next generation of maybe researchers, but also science enthusiasts, so people who don't dream of a career in the lab, but who just want to kind of watch from the sidelines?
So first of all, caveat, I'm not a neuroscientist. I come from molecular biology. So I think today, first of all, if you're passionate about science, it's amazing. We have so many resources, and with open science, a lot of the latest research is available almost always, almost as soon as it gets published. So it's really an amazing time to be alive, and a lot of science is moving to the computer to do analysis on huge data sets that are available. So if you're passionate about it, you can just call your local university, find people where you can volunteer in their lab and see if this is something that's for you. And you can, if you have computer skills, there's a ton of people that will be really happy to help you and guide you to find and answer questions that are interesting to you. And just.
It's funny, I think a lot of the elements that you touch upon are also kind of things that we're trying to encourage or promote in DeSci, right? Like, okay, you have an interest. Where do I get started? Okay, you can join maybe like a Reddit thread, or you join a Discord group. You start asking questions. How can I get involved? Is there, maybe is there a paper that I can review, or can I analyze the data set for anybody? So I think probably just like in most things in life, right? Like, it's just a matter of getting started, asking the right questions, and kind of like finding your group. So
yeah, and I think anyone that's willing to put the time and effort can do it. Especially today, it's the barriers are becoming lower and lower, and we appreciate skills from different group sets. Like in biology today, we try to work with everyone because my background, I didn't start with biology, we didn't get to that, but I actually learned physics because I didn't think biology is serious enough, just as a foundation. And it's changing, it's really changing. They're teaching a lot of computer science to biologists today. We need people with skills in chemistry. We need people that can do field work, collect samples, work with people. There's, of course, the more humane side. I'm less connected to it. It's really important. It's just not something that I'm particularly good at.
Speaking of things you're maybe not particularly good at, I actually really love this kind of initiative or like thread that I've seen on social media. I don't know if you're familiar with it, but it's called Overly Honest Methods. And it's where people kind of explain like incubation step for 24 hours in brackets. I left the plate out of the fridge or something like that. So I'm curious if you wouldn't mind divulging maybe one of your overly honest methods, or something that kind of happened to you maybe early, of course, early in career, because you learn and you mature as you go. But is there anything you could share with us?
So one of my favorite ones is I did my PhD on nematodes because you can really ask amazing questions there and get accurate answers relatively quickly. And you're done with your worms. You maybe created a new strain. You want to freeze them. It's a great property. You take your worms, you put them in the freezer, and in 50 years, if someone wants to, they can come out and take them and keep working on them. And then you have the freezing medium. And it's an elaborate thing with trace metals and stuff, and you're looking at it and like, they're sitting in the freezer. Why do they need trace metals for? They're not metabolizing anything, they're frozen. Let me try it with M9. It's the equivalent of tap for worms. It works just the same. I'm trying to picture, like, okay, someone grabbed that bottle from their bench 50 years ago because that's what was there, and it worked for them, and we're all doing it for no good reason. So yeah, if you can cut the protocol in half, I'm always for that. But the overnight steps, yeah, people want to get home, so they'll always put their samples overnight.
Awesome. Well, thanks so much, Danny. We actually do have a worm project going on, which will make you aware very soon. But yeah, we're a big fan of nematodes and C. elegans over here as well.
It's the best model organism.
Could not agree more. Could not agree more. Anyway, we'll talk soon. Thank you so much for coming on. Great conversation. Great conversation. Appreciate you answering all the questions. And yeah, if people want to fund Dr. Barr's research, they can check out the project on Catalyst and learn more there. But yeah, with that, we'll let you get back to your research. And yeah, thanks so much for joining us.
My pleasure. If anyone wants to write questions later and ask, I'll be happy to answer.
Awesome. Thank you. And with that, we'll bring on Michael Torres, who's going to give us an update on Artan Bio. But yeah, thanks, Danny.
Thank you.
Cool. Hey, guys.
What's up, Michael? How are you?
Hey. Hello, hello.
Hello, hello.
Yes.
Good to see you. Anyway, for those not aware, we're bringing on Michael Torres. He's the CSO, or is it CEO or CSO?
Anthony and I just kind of said, well, I'm already CEO of one company. I said, okay, let me make sure my investors don't go too crazy on my other company. So I'll take CSO.
Okay, it's a power struggle. It's a power struggle. You could say it.
Yeah.
And he won.
Yeah. If he won, he'd be here. So kidding.
True.
He's a good guy. I love Anthony. Maybe he's watching. I don't know.
Who knows?
Shout out to Anthony.
Yeah.
Shout out to Anthony. Cool. Well, yeah, thanks so much for joining us. I know you got a busy schedule. I'm sure many of the people do as well watching. So let's jump right in. So yeah, Artan just had a big data release. You guys got data back from the contract research organization that you're working with. I thought, there's so many other videos that people can go watch, like the full background on Artan. But if you don't mind giving like a 1-minute setup just to, get people up to speed with where you guys are now, and then we can get into the data.
Yeah, no problem. So when it comes to Artan Bio, what we're focused on is sort of this interesting aspect of biology where, there are many proteins that are involved in things that sort of prevent or slow aging related processes, like things to keep our DNA like stable, intact, no mutations, things that help suppress, cancerous cells that may come up in the body. These are called tumor suppressors. And so what we noticed is that over time, these proteins accumulate mutations that then sort of dampen their ability to do their job correctly. And so as those things start to come down, you're more susceptible to genomic instability, to premature aging, to neurodegenerative diseases, et cetera. So we came up with a strategy to, deliver an agent that could, the idea is to suppress those over time. And if we suppress those mutations, perhaps these, all of these cellular processes will stay stronger for longer. And then, that could be a really effective way to significantly increase our lifespan, because we're not touching on one aspect of aging. We're touching with multiple aspects of aging with our therapies. So that's what we're working on, and that was what was funded by VitaDAO, and we continue to fund with a molecule platform with the tokenization of this project, Vita RNA. So cool. So yeah, I mean, if you want me to hop right into the data,
let's do it. Let's waste no time at all.
Cool. Yeah, let me just share my screen real quick, and let me. Okay, excellent. Let me give you guys the right screen, and hopefully this is coming up right now. Does that look good? Do you see that? Or maybe Benji, you've got to allow that to.
My computer's.
Okay, perfect.
Computers, you're good? Amazing.
It looks like it's good, right? I can see it. So you guys can see the slides?
Honestly, yeah. It looks great.
Great.
Yeah, I had the power. I added it.
Logan. Okay, great, cool. Right. So what I just said, right? There's the hallmarks of aging. If you look on the right side, the primary types of aging are genomic instability, epigenetic alterations, loss of proteostasis or proteins, and then various other aspects. And so the take home is this genomic instability underlies many causes of aging. And so unlike other approaches that focus on a single aspect, we're trying to, again, address this really interesting biology that impacts many proteins and processes involved in the primary causes of aging. And so what that actually means is we're focused specifically on a type of amino acid that's called arginine. And so DNA makes RNA and makes proteins, and proteins are what, does functions in the cell. And so, these arginine is the DNA code. There's all of these triplets called codons that can code for arginine, but one in particular, the CGA codon, gets frequently mutated. And if it gets mutated, the C, it can turn into a UGA, and that's actually what's known as a stop mutation or stop codon. And so if you look at, various proteins in our bodies that have, this arginine CGA codon, there's many evolved, as I said, in age-related proteins like, a DNA damage, response proteins like ATM, Bloom syndrome helicase, involved in, replication stress. So again, really important conditions. And then importantly, it's not just at the DNA level that these mutations occur. And if they occur in the DNA level, they can cause diseases like cystic fibrosis, perhaps even autism, neurological conditions like Alzheimer's, we just heard about, or again, in the case of tumor suppressors that suppress tumors or cancers, these are very important proteins. And so again, as I mentioned, these mutations can occur, and they also can occur at the RNA level as well. So actually in all of our bodies, like right now, we're actually like, these mutations are happening, and sometimes it happens in a tumor suppressor and you get cancer. And so again, like we're sort of focused on figuring out a strategy to suppress those mutations, as I mentioned. And so with money that we received from VitaDAO, we then designed some vectors that can express a different type of RNA molecule that will then sort of correct that mutation, as I said, or in this case, we call it suppress these arginine nonsense or stop mutations. And so, in order to test that, we took advantage of cancer cells that have a nonsense mutation, an arginine mutation in P53. P53 is called the master regulator of the genome. I forget, there's other sort of names. It's a very important protein that, like elephants, as an example, have multiple copies of P53, and that's thought to, be one of the reasons why they have lower cancer incidence. But point is, it's a reporter system for us, so something that we can test in cells that, as we design these vectors, what we're looking for then is, in the case of like lane 1, there's no transfection, so these cells are just growing normally because they have that stop mutation, which causes the protein and mRNA to stop. You don't see any protein here, as an example. But in the case of lanes 4 and 5, which are two vectors that we designed to express this RNA molecule to rescue P53, as you can see, with these two sort of dark bands that have emerged, that shows that we actually can rescue these types of arginine mutations that are occurring in many proteins. But again, we're using this P53 as a readout there. So again, this is taking sort of a type of RNA, a DNA called the plasmid DNA that we can transfect or put into cells that then will express this RNA. But for most cases, that's not a viable way to deliver this molecule into humans or for animals, and to hit many tissues, because again, this is happening all over the body. So ideally, we'd want to hit as many cells as possible. And so the way to do that then would be to then formulate this drug into an AAV, so a gene therapy vector. And in this case, we chose AAV9. And so, like in this schematic here, you can put your gene of interest, or in our case, it's this arginine suppressor. We put that into the virus. We make the virus, and then the virus then can infect or transduce cells. And in the case of people who are missing like a gene, for instance, like SMN, which causes SMA, spinal muscle atrophy, there are approved drugs on the market that use AAV9 in order to rescue that disease. And so that's actually been very effective model. But the point is, AAV9 is a validated vector to deliver our suppressor. And so with that, then we then went back to our contract research lab to ask them to formulate our vector into AAV9. And then with that, then that process, if that works, can now really sort of put us on track to scale up the virus to then support getting into humans, which is ultimately what our goal would be. So what we did then, and this is kind of what the most recent news was, is we took these two vectors where we see work just as naked DNA, and we put it into a lentivirus, and then after we made that, then put them back in these cells to see if they can rescue protein. And so in this case, this is what this slide shows here. So it's these same model cells that I mentioned. And here on the left, we took what's called GFP, green fluorescent protein. So this is a reporter. It just makes a protein that fluoresces, so you can see it easy in the microscope. So instead of delivering our suppressor, it delivers GFP. And so you do this first just to make sure that these cells can be infected with the AAV9 virus. And so that's what that shows. You can see these green cells now. That means that the virus went into the cells. and expressing GFP. So over here on the right, it's that same assay, which is called a Western blot, where now we took our 2 lead vectors, made them into viruses, and then tested the activity here. And I won't go into the details specifically about the differences, but as you can appreciate, there's one virus that clearly makes more protein than the other. So this, what we're calling Artan 102, now is something that could be a preclinical candidate that we could scale up to start moving into animal studies, and ultimately, into people once we, make it, manufacture it, show that it's positive, and show that it's, safe for human use. And again, with that, we'll be able to do animal studies, and not just animal studies for safety, but in this case, we can look at a variety of different models, like again, we mentioned cystic fibrosis, but there's other diseases in which arginine could be important. And so, but again, the key is to make enough virus in a way that's safe for animals, and then ultimately safe for humans. So then you've got to start working with manufacturers who can do that. And so in this case, we have an example of like Lonza, who we've already started working with, who can, Lonza is one of the leaders in making cell and gene therapies for companies. And so, this could be, this is one of the next steps for us, is to work with the manufacturer, make a lot of high quality virus, and then we'll have a bunch of it that we could start testing in animal models. So, which is exciting. This is like, on path to creating a drug that's doing something interesting in biology that could address aging and be important for rare disease as well. And this is all being funded by the community. And we're starting to drop data in DeSci labs and have these repositories, and, want to do this very openly and collaboratively with the community.
So cool to hear the updates, Michael. Thanks so much for sharing. And if you can go back to your previous slide, it's so clear here. For maybe people who are not so familiar with Western blots, like more is more. So the darker the band, the more the protein. And it's clear that you have like, one band here to rule them all. So pretty exciting that you were able to get to this result so quickly and so effectively as well. So huge congratulations, obviously, to you and the team for the hard work into this research so far. And maybe it's actually kind of funny timing, because obviously we had Daniel Barr on earlier, who said, exposure to viruses will increase your risk, of dementia later. And here now we're talking about, we're actually going to use this virus to come back and save mutations in P53. So just wondering maybe if you could tell people not to be afraid of the viruses, that these ones have been made harmful, kind of like how that process works and why we would actually want these ones to get into our body.
Yeah. So, we have, most of us have been infected with AAVs and other virus. We're constantly, sort of being exposed to viruses, and some live longer in our bodies, like, herpes and, HPV. And in the case of HPV, it's associated with cervical cancer, right? But viruses, as any of us who've gotten colds or who had COVID, they're really good at affecting multiple parts of our body. Okay. And the way that they do that, right, is by actually getting, in most cases, getting inside cells, where then they replicate, they hijack the cellular machinery. So they're really good at, they're professional makers of material. In their case, they make it for themselves to then, replicate and all that. But in our case, we actually can use them to replicate things that are important for the human body itself, and not just for the virus. So in the cases of these viruses, they have basically just the minimum components to get in cells, but they don't sort of replicate on their own and cause their own sort of diseases or disorders that other viruses can get. So it's actually a really unique way to deliver medicines, basically that can be very long lasting, which is the important aspect for this, is that this might be something that we would want to chronically suppress for as long as we're alive, and by doing that, again, we could have significantly longer lifespan, and obviously this is early, so we don't know really what the benefit could be, but we're nobody's tried this before, so we're optimistic that, hey, this could be really effective.
I mean, somebody's got to try it, right? Somebody's got to be the first. So
and why not crypto and DeSci and the whole thing? I mean, I think that's just it is really cool to kind of see this play out.
And I'm also curious about, so earlier you talked about the different hallmarks of aging and how these types of mutations are present across different proteins, across different functions that all kind of play into the aging phenotype, or the progression of aging that we will all face. And the current therapy that you're developing, so Artan 102, is this going to be something specific for p53 mutations? Is this something that's going to be broadly applicable to all different types of mutations? Are we focusing on one, or is p53 more just an example, kind of like use case at this moment based on the cell type and the kind of mechanism that you wanted to show?
Yeah. So that's a good question. I mean, the high level answer is we think this is broadly applicable and is likely working across any of the CGA codons and that could be present in multiple different cells and what have you. But yes, p53 today is a reporter for us, so something that just shows us that this concept can work. At the same time, it could be a very important therapy for p53. I mean, people have been trying to figure out how to sort of either drug p53 or sort of figure out how to increase protein levels, because again, there is this data out there in species, et cetera, that p53 is really important for suppressing cancer, and cancer obviously can cause premature aging. So but I think the more sort of interesting question, and this is what we wrestle with at Artan a bit, and then we're going to talk to the community about this stuff too, right? Is there is a strategy where you do go after a rare disease to get your patient data and get buy-in from FDA. And so you focus on a specific disease. And then with that data, you can sort of use that to then go broader and broader. And as FDA gets more familiar with like this drug, and you have safety data from various patients, it shows it's working, it's safe, like you could expand into like broader indications, or aging is obviously kind of like the biggest indication there is, and so I think internally we probably will start with a disease model that has a single mutation in arginine that causes like Dravet syndrome. It's like a seizure syndrome, for instance, where arginine is important. I mean, I skipped over it, but I think it is important to for people to understand that, as I said, there's these stop mutations that can occur, in different proteins, but there's also different types of stop mutations that can occur, because different amino acids can get mutated and they can cause, actually, there's 3 flavors of stop mutation. So it's a whole biology thing we could, chat about. But if you look across, all of the different disease-causing nonsense mutation, arginine is actually the most common nonsense mutation across all of these different disease types. So arginine is a really important, going after arginine, this approach is really important. And there are actually they're rare diseases, but there's large populations where arginine would be very effective. So there is that additional strategy where arginine could, if you said we're going to just be agnostic to the disease, but we're going to just go after any disease that has an arginine mutation, stop mutation that causes it, this therapy could actually be for all of those different diseases as well. And so that's why, again, I think it's actually pretty pretty exciting.
So nice to be able to have choice, right? Where you can kind of start broadly and just follow the data as it comes to be able to make better choices along the way. But yeah, so interesting that arginine is particularly susceptible to these types of mutations. And maybe before we run out of time, so obviously you have some great data in hand. You showed your plan for next steps, working together, with Lonza, scaling up the manufacturing. Maybe what would you want people to know about the next steps in terms of expectations or timelines, or what you want to communicate, or maybe things that you have cooking in the background and updates to share?
Yeah. So, we're literally on calls with CROs, these contract research labs today. We had a call with Jackson Labs, and Jax makes a ton of mouse models, and they also have aging models as well. So we're going to start, putting out some thoughts around models and sort of the next step for in vivo, mouse studies. And so whether it's in our Telegram chat, or other channels, like there are places to get insight and to contribute. I think we try to also obviously incentivize token holders to participate in these conversations as well. So as a reminder, we raised $300,000 through the first Vita RNA auction. And we there was a proposal to unlock the first 100,000, and that's what has continued to fund this work to date. Now again, on the manufacturing side, I mean, Lonza's very expensive. I got a proposal. I mean, we probably can share, but it's like almost close to $1 million if we want to use like Lonza's like stable cell line. There's some technical stuff, but or it's around 300, 400K. So these things aren't cheap. We have 200K that we can submit, that we've already raised to unlock. So we have to think through strategy. Okay, maybe we don't go with Lonza. We go with like someone else who can make it to do some of this early proof of concept work, and then try to raise additional funds to do that. Work with Lonza, however, or we bite the bullet and say, hey, look, we're going to like go all in and fund this and see how far we can go with, continuing to unlock, capital and resources to move this forward. But yeah, there will be a proposal, coming out soon to talk about, to formally, take to the token holders to say, hey, we want to move this forward. And here's our thought, and we'd love to get your, buy-in as well on that. So.
Or if anybody has a Lonza discount code, feel free to drop it in the chat.
There you go. And we are going to try very hard with them. It's a new platform. One of theirs is a new platform, and they had a mark. Well, we're looking for beta testers. I'll say, okay, well, slash that, make that 50% off for us, or maybe take equity or buy the token themselves. I don't know. Like let's get creative here and really leverage what we're doing.
I love the creative thinking, Michael. I don't think that's in short supply here.
Nice. Cool.
Awesome. Well, thank you so much for joining and giving the update on the data and the progress at Artan Bio. So if anybody's interested in following along, you can check out the website. You can find them on the VitaDAO website. They also have their own website, Vita RNA. The token is also, or information about the token is also there. But yeah, really loved having you on, and looking forward to the next announcement and the next steps.
Excellent. Thank you. All right.
Thank you so much.
Bye.
Hi, everyone.